பயாலஜி இன் தமிழ் கோர்வை சேனல் சார்பாக அனைவருக்கும் வணக்கம். என் பெயர் கண்ணதாசன். In this video, I am going to present Classical Genetics Part 5. Part 1 to Part 4, the links are given in the description. I request all the students to please go through all those videos so that you are able to easily understand this video. In this video, we are going to learn 2.3.7 extensions of Mendelian Genetics Gene Interaction, 2.4 Intragenic Gene Interaction, 2.4.1 Incomplete Dominance, and CBSE part of the Incomplete Dominance, 5.2.2.1 Incomplete Dominance of the CBSE book. This video also going to be accompanied by one Google Doc form question paper. It will be very useful for both NEET student as well as board student. The 1 mark MCQ, you can get the immediate result once you finish the test. And the descriptive answer, it will be very useful for board student. Board student can write it in a separate notebook and do the self correction. So I request all the students to take the textbook, go to the particular page, notebook and also pen, and each slide carries many important information. You please take note of it. It will be very useful for you in the examination. At the time of the examination, during the revision, it will be very useful. So please take note of it. Now let us enter into this video. This is the subtitle 1, 2.3.7 Extensions of Mendelian Genetics. Extensions, it means Mendel's law of dominance, law of segregation, law of independent assortment. All these having their own exceptions. These exceptions are said to be the extensions of Mendelian genetics. I hope you would have understood what is the meaning for extension. The exception, it means Mendel's law of dominance, law of segregation and law of independent assortment. They are not universal in their application. There are some exceptions. Those exceptions are studied under the extensions of Mendelian genetics. Exceptions to Mendelian principles. Straight away they are telling it. Apart from monohybrid and dihybrid, trihybrid crosses, there are exceptions to Mendelian principles. That is the occurrence of different phenotypic ratios. Mendel obtained the phenotypic ratios 3.3:1 for phenotypic ratio, genotypic ratio 1:2:1. In the same way for dihybrid cross, Mendel obtained the phenotypic ratio 9:3:3:1. But when other scientists tried the same experiment in other plants with respect to other characters, they could get entirely different result. That is what called as exceptions. Exceptions to Mendelian principles. That is the occurrence of different phenotypic ratios. The more complex pattern of inheritance are the extensions of Mendelian genetics. Here you can see there are examples where phenotype of an organism is the result of the interactions among the genes. You can see the genes in the form of alleles. Capital T is an allele for tallness and the small t is the allele for the dwarf. When they interact, tall is expressed. So this is called as the allelic interaction, or the interaction between the genes which occupy the same gene locus. Same gene locus means they are called as alternative form of the same genes. They are called as alleles. Subtitle 2, Gene Interaction, we are going to learn. A single phenotype is controlled by more than one set of genes, each of which has 2 or more alleles. Now you can see this is one gene and this is another gene. Both the genes may be responsible. Here I am giving you an example with a picture for you to understand the two genes involved in the expression of one character. Understood? And this one character consists of two variation. Understood? So usually we learnt in Mendelian genetics one gene with two alternative form. It means alleles responsible to produce one character with two variation. But here two genes with their own allelic form. A is a gene with its allelic form A small a. In the same way, capital B small b. These are the two different genes occupying the two different gene locus. They interact to produce one character and their variations also. So this is called as gene interaction. This phenomenon is called as gene interaction. So the question can be asked, what is gene interaction? Right? So sometimes two or more genes responsible to produce only one character with two or more variation. This is called as gene interaction. Many characteristics of the organism, including structural and chemical, which constitute the phenotype, are the result of interaction between two or more genes. Many of the characters, including the characters of the human being, there the character is due to the gene interaction where two or more genes involved. Suppose you take the human skin color, human height, human weight. Here many genes are involved. So that is due to the interaction between the many genes. And remember this concept, these genes are present in a different locations, different places. They are not occupying the same gene loci. Alleles only occupy the same gene locus. Now you take this gene consists of its own allele. They are occupying the same gene locus, and this is another gene which is having its own allele on the same gene locus, but both the genes are occupying the different loci, different location on different chromosome, but still they are responsible to produce the particular character with two or more variation. So this is called as gene interaction. Mendelian experiment proved that a single gene controls one character. Single gene, it means T gene, responsible to produce the height of the plant. But in the post-Mendelian findings, various exceptions have been noticed in which different types of interactions are possible between the genes. Different types of interactions are possible between the genes. This exception to Mendelian principle, the gene interaction concept, was introduced and explained by W. Batson. So Batson's factor or Batson's hypothesis can be asked in 2-mark question. This concept is otherwise known as factor hypothesis or Batson's factor hypothesis. This can be asked in 2-mark question. According to Batson factor hypothesis, the gene interactions can be classified as intragenic gene interactions. You see gene interactions. Draw the flowchart of various types of gene interaction can be asked in 3-mark question. So this is the beginning of the flowchart. The flowchart continues. So there are two types of gene interaction. One is intragenic gene interaction or intraallelic between the alleles of the same gene, then it is called as intragenic gene interaction or intraallelic or allelic interactions. And another one type is intergenic gene interaction or interallelic or non-allelic interaction. Non-allelic, they are not alleles. What are alleles? Alleles are the alternative form of the same gene occupying the same gene loci. When the alleles of the two genes not occupying the same gene loci, then it is said to be non-allelic interaction. This is the subtitle 3 2.4 intragenic gene interaction. We are going to concentrate on intragenic gene interaction. Intragenic within the gene, intergenic between the two genes. That is the difference. Intragenic within the same gene, it means allelic. Intergenic, between the two different genes occupying the two different location of the different chromosome or same chromosome, then it is intergenic gene interaction. I hope you would have understood. Now 2.4, intragenic gene interaction. This can be asked in one 5-mark question. Interaction take place between the alleles of the same gene. Now these are the two alleles of the same gene. Now this is one of the simple Mendelian example for intragenic gene interaction, where capital D interact with small t to produce the tallness. Then it is called as the intragenic within the gene where the alleles occupying the same gene loci at the same gene locus is called as intragenic or intralocus gene interaction also. So what is intragenic gene interaction or intralocus gene interaction can be asked in 1 2-mark question. You please take note of it in your notebook. Now the flowchart continues for intralocus or allelic interactions, right? So between the alleles of the same gene occupying the same gene locus, then it is said to be intralocus interaction or allelic interaction, in which you see these are the types. This can be asked, as I already told you, 3-mark question. Number 1, dominant relationship, complete dominant, example tall and dwarf in pea plants. Incomplete dominance, codominance, overdominance. So these are the dominant relationship between the alleles. Dominant relationship between the alleles. Next one is second category, lethal genes. Lethal genes means in one form of the allele responsible to cause the death of the organism, then it is called as lethal genes. Lethal gene always found to be fatal. It causes the death of the particular individual possess the particular allele. Example, dominant lethal. Dominant lethal means the dominant allele may be lethal in nature. Recessive allele may be lethal in nature. Then recessive lethals, conditional lethals, sex-linked lethals, balanced lethals. Then third category is the multiple alleles. So these are the various categories of intralocus interaction or allelic interaction. Intragenic gene interaction, we are going to study in state board book 4 categories. One is incomplete dominance. In this video we are going to completely cover incomplete dominance with respect to state board and CBSE book. Codominance, multiple alleles, pleiotropic genes are common examples for intragenic interaction. So first we are going to cover 2.4.1 in state board book, incomplete dominance. Here no blending of genes take place. No blending of genes take place. Incomplete dominance can be asked in one 5-mark question. The German botanist Karl Correns 1905 did experiment in 4 o'clock plant. So this is the flower, 4 o'clock flower. It's a seed, and the plant, it is a herb, 4 o'clock plant, and its scientific name is Mirabilis jalapa. Mirabilis jalapa. Here you can see the two colored flowers. One is red color with the genotypic symbol capital R1, and for white the genotypic symbol capital R2, capital R2. This is the parental generation, where you could see the genotypes are homozygous, capital R1, capital R2, capital R2. When they are crossed in the F1 generation, they produce heterozygous alleles, capital R1, capital R2. This is the F1 generation. When the pure breeding homozygous red, capital R1 parent is crossed with homozygous white, capital R2, the phenotype of the F1 hybrid is heterozygous pink. This is the heterozygous pink. It is neither it is giving the appearance of the red parent nor giving the appearance of the white parent, but an intermediate color is produced with respect to the heterozygous genotype capital R1 capital R2. The F1 heterozygous phenotype differ from both parental homozygous phenotype. It differ from both parental homozygous phenotype. This cross did not exhibit the character of the dominant parent but an intermediate color pink. So actually what they expected, as Mendel could get any one of the character either red or white will be produced with the F1 as against their expectation. It has produced an intermediate color, that is the pink color in the F1 generation. When one allele is not completely dominant to another allele, so red is not dominant over white or white is not dominant over red. So in such case, it shows incomplete dominance, the pink color flower. Such allelic interaction is known as incomplete dominance. F1 generation produces intermediate phenotype pink colored flower. When pink colored plants of F1 generation was interbred, it means selfed. Selfing only take place during the F1 generation. Okay, with respect to the Mendelian experiment, they also followed the same pattern. When the selfing of F1 plant occurs in F2, both phenotypic and genotypic ratios were found to be identical. They are 1 is to 2 is to 1, 1 red, 2 pink and 1 white. Genotypic ratio also found to be the same, 1 capital R1 capital R1, 2 capital R1 capital R2, 1 capital R2 capital R2. So this is the ratio which is against Mendelian ratio of the monohybrid cross, where the phenotype is 3 is to 1. Phenotypic ratio is 3 is to 1. But here phenotypic as well as genotypic ratios stand 1 is to 2 is to 1. And here this is because of the production of an intermediate character by the heterozygous genotypes. You could see here capital R1 red and capital R2 white, like parental genotype, parental phenotype, whereas capital R1 capital R2 the heterozygous 50% of the plants become an intermediate character. So that is the reason why the ratio, Mendelian ratio, deviates from 3 is to 1 to 1 is to 2 is to 1 in 4 o'clock plant. From this we conclude that the alleles themselves remain discrete, unaltered, proving Mendel's law of segregation. Here Mendel's law of segregation take place. Remember that Mendel's law of segregation take place. The capital R1 segregates from capital R2. That is the reason why in the F2 generation we are able to get both the parental character, that is the red as well as the white. Understood? Along with the intermediate character also, that is the pink. So what is taking place? Segregation taking place. The alleles become discrete, unaltered when they stay together in F1 generation. That is the point to be noted. When they are staying together in the F1 generation, they stay together without mixing, without blending. That is the reason why it is called as no blending occurs between the alleles, because they are discrete units, independent units. They are able to segregate themselves when the F1 generation plants produce the gametes. So R1 segregate from R2, R2 segregate from R1. That is the reason why, students, it is able to produce the red color flower individually and white color flower individually, and 50% of the flower found to be pink, because the R1 and R2 stay together again in 50% of the plants. The phenotype and genotype ratio are the same. It means 1 is to 2 is to 1, and there is no blending of genes. If there is a blending of genes, what we would have got in the F2 generation means all pink. We would not have got red, the parental combination or parental character, or white, the parental character. How we are able to get? Because the genes are not, alleles are not blending. So there is no blending of genes. In the F2 generation, R1 and R2 genes segregate and recombine to produce red, pink and white in the ratio of 1:2, 2:1. So R1 in the F1 generation, R1 segregates from R2. Okay, they R1 segregate and combine with another R1 to produce red. In the same way, R2 segregate from R1 to combine with another R2 produce white. But still 50% of the alleles or 50% of the genotypes carry the heterozygous nature, that is responsible for the pink color, 50% of the pink color flower. R1 allele code for an enzyme. So they are giving the biochemical explanation. R1 responsible to produce an enzyme which is responsible for the formation of red pigment. So the R1 responsible to produce an active enzyme, students. This active enzyme convert the pigment, inactive pigment, into active red pigment. So it is responsible to produce the 50% red. R2 allele code for defective enzyme. So no red pigment is produced. So 50% of the red pigment and 50% of no red pigment, it means white, that is responsible to produce the intermediate color pink. 2 R1, capital R1 are needed for producing red flowers, because all the enzymes become 100% enzymes become active, so they are able to produce 100% pigment into red pigment, so red color flowers are produced. Whereas capital R2 genes needed for white flowers, where the enzymes are not active, 100% enzymes are not active, so no pigment is converted into red pigment, so all the flowers become white in the case of capital R2. If blending had taken place, so they are giving a condition. If the blending had taken place between the capital R1, capital R2, the original pure trait would not have appeared in the F2 plants. Okay, F2 plants would have pink flowers. They have only pink flowers. So no blending of alleles take place, and the alleles segregate because they are discrete units, so they are able to produce the red color parental character and white color parental character. It is very clear that Mendel's particulate inheritance taken place in this cross. So law of segregation is taking place, but law of dominance is not obeyed. Law of dominance is not obeyed, whereas law of segregation take place. That is the reason why the pure red color flower and pure white color flower produced, and 50% that is heterozygous genotype produce pink color flowers. So it is clear that Mendel's particulate inheritance taken place in this cross, which is confirmed by the reappearance of original phenotype in F2. Here, the explanation to the intermediate heterozygotes phenotype. How are we going to interpret the lack of dominance? The pink color is produced, that is due to the lack of dominance, and give explanation to the intermediate heterozygote phenotype. How will you explain incomplete dominance at the molecular level? So the explanation is given in the state board book. Gene expression is explained in a quantitative way. In a quantitative way, we are able to explain this. Wild type allele, capital R1, which is a functional allele, when present in two copies, capital R1 produce a functional enzyme which synthesize the red pigment. So this active enzyme produced by capital R1 allele responsible to produce the red pigment. The mutant allele, it means R2 produce, it is the defective allele due to some reason mutation taken place in R1, so it is converted into R2. it is producing an enzyme which cannot synthesize necessary red pigment. The white flower is due to the mutation causing complete loss of function. So no red pigment is produced. The F1 intermediate phenotype heterozygote R1, capital R1, capital R2 has one copy of the allele capital R1. So 50% of the functional protein resulting in half of the pigment of red flowered plant, and so it is pink. Only 50% of the enzyme or 50% of the pigment protein can be converted into red. Another 50% is not converted into red, so it has become pink. The intermediate phenotype pink heterozygotes with 50% functional protein is not enough to create the red phenotype homozygous, which makes 100% functional protein. So the 100% functional protein produced only by the homozygous, not by the heterozygous condition. Now this is the incomplete dominant from CBSE book, where CBSE book entirely different examples given and different interpretation given. It is very useful for the NEET aspirant of the state board as well as the CBSE student. When experiment on peas were repeated, it means Mendel's experiment repeated using other traits in other plants. It was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in dog flower. So the dog flower, snapdragon or Antirrhinum is an example given in the CBSE book. Is a good example to understand incomplete dominance. So here you could see the red color and white color flower and also pink color flower. Here you could see in a cross between true breeding red flowered, here you see the genotype given capital R and for white smaller as as against in the case of state board book where capital R1 capital R2 capital R2. So in the NEET question you may not get the same type of genotype capital R1 capital R2 capital R2. Instead of that, even in the incomplete dominance they would give capital R for red and small r for white. So in the F1 generation, capital R small r become pink. So that is what you could see here. This is the parental generation where you could see red color flower capital R, white color flower small r, and the gametes become capital R small r. In the F1 generation, all pink capital R and small r. Again, when the F1 was self-pollinated, the F2 resulted in the following ratio. It means 1 capital R red, 2 capital R pink, 1 small r white. So here you could see in the Punnett square all pink color flowers when self-pollinated, we are able to get 25% of capital R homozygous red and 25% of small r homozygous white and 50% of heterozygous capital R small r become pink. So the phenotypic ratio and genotypic ratio become the same. Are you clear? As against the Mendel's phenotypic ratio 3:1. Are you clear? Here the genotypic ratio were exactly as we would expect in any Mendelian monohybrid cross. It means genotypic ratios stand same with respect to Mendelian monohybrid cross, but the phenotypic ratio had changed from 3:1 dominant versus recessive ratio. So that is what we are able to understand from here. 1:2:1 phenotypic ratio in the place of 3:1 genotypic ratio stand same. Here you could see instead of two colors red and white, one more is added. it means the pink, the intermediate character is added. Whereas in Mendel's monohybrid cross, yellow is dominant over green, we learnt, and no intermediate character is produced. In a dominant character, yellow is produced, recessive character, green is produced. That's all. Okay, but here, in addition to that, one intermediate character is produced. So that is the deviation from Mendel's theory, Mendel's law. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. So every gene is responsible to produce a particular trait. What is gene? A segment of DNA which is responsible to produce a particular trait. In a diploid organism, there are two copies of gene, that is as a pair of alleles. So these are the pair of alleles. Now these two alleles need not always be identical. These two alleles need not be always identical as in the heterozygote. So one may be wild and another one may be a mutant. It means genetically it is changed. One of them may be different due to some changes. So it would have undergone some changes, mutation would have occurred, so it has become R2. So R1 able to produce enzyme, R2 not able to produce enzyme. So the mutation modified the R1 into R2. Let's take an example of a gene that contains the information for producing an enzyme. So R1 is able to produce an enzyme, whereas it has undergone mutation and become R2. R2 is not able to produce the active enzyme. Now there are two copies of this gene, the two allelic forms. So these are the two allelic forms, one is wild and another one is mutant. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate. It means the pigment precursor into the product, that is the red pigment. So the product becomes the red pigment due to the presence of an active enzyme. Theoretically, the modified allele, it means R2, could be responsible for the production of. So these are the three possibilities given in the CBSE book. It will be very useful for the NEET aspirant of State Board as well as CBSE. First condition, first possibility, the R2 also produce the normal enzyme or less efficient enzyme. Possibility 1. Possibility 2. The R2 produce non-functional enzyme, that is the mutant allele produce non-functional enzyme. This is the possibility 2. Possibility 3, no enzyme at all, the R2 not producing any enzyme. So this is the possibility 3. In the first case, the modified allele, though it is modified, it is able to produce the enzyme which is like the enzyme produced by unmodified allele. So this is the possibility 1, where we could not find any difference between the expression of R1 and R2 because both produce the normal enzyme, so both convert the substrate into product. So that is, it will produce the same phenotypic trait. So we are not able to identify which one is R2 and which one is R1 because both produce the normal enzyme. Even after the mutation, there is a possibility, okay, because there may not be any change in the amino acid sequence. So the same enzyme may be produced. That is the possibility 1. The result in the transformation of substrate, yes, such equivalent allele pair are very common. It is very common in nature. But if the allele produce a non-functional enzyme or no enzyme, non-functional or no enzyme, the phenotype may be affected. It means the enzyme produced by R1 convert the substrate into product, whereas R2 not able to convert the substrate into product. That is the reason why the red color pigment is not produced, only white color flowers are produced. The phenotype or trait will only be dependent on the functioning of the unmodified allele. Unmodified allele. So it is modified as mutant. So it is not able to produce the active enzyme. So it is not able to convert the inactive red pigment into active red pigment. Only white color flowers are produced. The unmodified functioning allele which represent the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. Generally, not always. Point to be noted. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. So the recessive trait weight is produced because of the non-functional enzyme from the modified R2 allele. So this is the end of this video, students. So far we learned extensions of Mendelian genetics, gene interaction, in which 2.4 intragenic gene interaction. We learned 2.4.1 incomplete dominance, where we learned when we are crossing red color flower and white color flower, we are able to get only the pink color flower, not any one of the character as dominant. So it is called as incomplete dominance. Still, we learned that no blending of genes occur. And with respect to CBSE, it will be very useful for state board NEET aspirant as well as CBSE student, incomplete dominant with respect to the different exams.